Hi everyone, I have no relationships or conflict of interest to disclose. I'm Corby Fink, and today I will be speaking to you about Nef inhibitors as adjuvants towards a cure for HIV/AIDS. Human immunodeficiency virus, HIV, is a complex retrovirus and is the causative agent of acquired immunodeficiency syndrome, or AIDS, and it contains the accessory proteins VIF, VPU, VPR, and Nef, with the latter playing a critical role in HIV pathogenesis. Combined antiretroviral therapy, or CART, is effective at prolonging the life of HIV-positive individuals by recognizing and destroying virus-infected cells in which the virus is actively replicating. However, HIV pathogenesis includes tissue reservoirs that contain latent or minimally replicative virus and therefore are not targeted by CART. One reactivation strategy is to purge this reservoir with the hopes of achieving a functional cure. However, with reservoir purging comes the expression of Nef that results in immune evasion and thus currently prevents a functional cure from being realized. Nef's pathogenicity is multifaceted and targets cellular pathways to subvert apoptosis, enhance viral replication, and suppress CD8-positive cytotoxic T lymphocyte, or CTL, immune responses directed at HIV. To suppress CTL responses against HIV, Nef, shown in pink in the bottom left figure, uses host protein packs to shuttle towards the trans-Golgi network, or TGN. Here, it interacts with Stark family kinases, or SFKs, which initiates a multi-kinase complex signal cascade that results in MHC class I internalization from the cell surface and sequestration to the TGN. This led us to the idea that if we conduct a screen to identify molecules that inhibit Nef-SFK interaction, we can prevent MHC class I downregulation, and thus these inhibitors could function as molecular adjuvants. An in silico drug discovery screen has previously been employed and successfully identified a Nef inhibitor termed compound 2C. 2C binds an imputative binding site of Nef identified by the black arrow in the upper right figure, and this binding allosterically inhibits Nef from interacting with SFKs. An additional in silico drug discovery screen was performed in an effort to identify novel inhibitors that function similarly to 2C. The novel inhibitor I will be speaking about is compound H31, and it was identified as a likely high-affinity inhibitor of Nef that could also help resolve toxicity issues that have been observed in the past. We predict that H31 can function as an adjuvant to prevent Nef-mediated MHC class I downregulation, shown by the red octagon in the bottom right figure that prevents Nef-SFK interaction. And by preventing this interaction, it can result in enhanced anti-HIV CTL responses to purge this viral reservoir. Our overall goal is to disrupt Nef-SFK interaction with H31 such that it functions as a molecular adjuvant. And to achieve this goal, we have three main objectives. First, we aim to determine optimal H31 concentration that prevents MHC class I downregulation by Nef in both primary and immortalized human CD4 positive T cells. We then sought to determine the optimal administration route in H31 concentration in an in vivo mouse model. And lastly, we plan to assess the role of H31 as an adjuvant in a dendritic cell or DC-based HIV reactivation strategy in a mouse model of an HIV/AIDS-like phenotype. To address our first objective, SEPT1 human CD4 T cells were infected with the lab-adapted NO43 pseudovirus that expresses GFP. After infection, these cells were cultured for up to 48 hours with increasing concentrations of either 2C or compound H31. And GFP served as a readout of this infection, shown by the upper right FACS plot, and within our infected or GFP-positive cells, we then looked at subpopulations that we based on low, medium, and high GFP expression. Surface MHC class I was represented as a fold change from uninfected cells in the summary graph shown at the bottom, following the same color scheme as in the FACS plot. And regardless of whether we saw GFP low, medium, or high populations, we observed an approximate 10% recovery in MHC class I with H31 compared to either NL43 infected cells or NL43 infected cells in the presence of inhibitor 2C. After observing partial surface MHC1 recovery, we wanted to confirm that these observations were Nef dependent. So we set up an experiment similarly to what I outlined previously, except for that we used both NO43 GFP and NO43 GFP delta Nef construct. In this way, we were able to look at surface MHC class I shown in the histograms for uninfected cells, black histogram, and infected cells, the red histogram. For NL3 containing Nef, we do see an improved MHC class I expression with increasing concentrations of H31, being the green and blue histograms, albeit not recovering to the level that we saw in our uninfected cells. In stark contrast with the histogram shown on the right, when the NO43 construct lacks Nef, there is still a downregulation in MHC class 1, but H3.1, even at its highest concentrations, does not improve any surface MHC class 1, as shown by almost complete overlap of these three histograms. So the take home from this is that H3.1 did rescue Nef-mediated downregulation and appears to do so in a dose-dependent manner, and that this observation was also consistent to what we saw in primary human CD4 T cells. We next moved into our in vivo mouse model of an AIDS-like phenotype using the CD4C HIV Nef transgenic mice, which have measurable decreases in CD4 T cells by 3 weeks of age and were generously donated to our group by Dr. Paul Joel-Coeur. This transgenic mouse was generated using a mouse CD4 enhancer outlined in red, a human CD4C promoter in blue, the entire NL43 coding sequence in green, and finally an SV40 poly-A tail in purple. It's important to note here that the upstream sequences dictate Nef expression in these tissues. So in this specific model, we have high Nef expression in CD4 positive and double positive thymocytes, peripheral CD4 positive T cells, as well as in macrophages and dendritic cells that are CD4 positive. This transgenic mouse model was chosen so that we could assess H3.1 functionality in vivo. Now, these mice are normally bred on a C3H background. However, we chose to breed these mice on a C57 black 6 background because it permits us to assess antigen-specific immune responses following vaccination. On a black 6 background, there is an MHC class 1 restricted peptide, commonly referred to as OT1 or synfecal peptide, and it's derived from chicken ovalbumin protein. This peptide in a vaccination model serves as a novel antigen to which a robust immune response is launched, and we can quantify this response by identifying synfecal-specific CTLs. So in the schematic at the bottom, we can immunize mice with dendritic cells presenting synfecal peptide, and once these cells reach the secondary lymphoid organs, they elicit a synfecal-specific CTL response that we can identify using tetramers and flow cytometry. We can also use the same strategy to determine the impact that our Nef inhibitor H3.1 has in the context of a vaccine strategy. Prior to vaccination studies, we first wanted to confirm that transgenic mouse model is functioning as expected. And then in this figure, the mouse model is just referred to as TG for ease of sight. What we did is we took splenocytes from either wild-type mice or transgenic mice. We cultured them with or without H3.1, as well as with or without synfecal peptide, and incubated them for time points that ranged from an hour up to around 6 hours. So in the top right FACS plots, we show a defined CD3 positive, CD4 positive population in wild-type mice, which is expected, and we see a noticeably smaller population in our transgenic mouse, which confirms that this AIDS-like phenotype is being represented properly. The graph that we have at the bottom summarizes our results, in which we measured the mean fluorescence intensity, or MFI, of synfecal peptide bound to MHC class 1, using an antibody that only recognizes this MHC class 1 peptide complex rather than either component on their own. And at each time point, 2, 4, and 6 hours, we measured this surface MHC class 1 for our wild type and our transgenic mice, with increasing concentrations of H3.1. So we saw consistent MHC1 synfecal complex in our wild-type mice, and this was consistent across incubation times and increases in H3.1. However, our CD4 T cells from our transgenic mice had improved surface presentation of MHC synfecal complex. So with these observations, we're in the process of conducting future studies that have a prolonged incubation time to see if this trend continues with a longer incubation time, as well as what appears to be a dose-dependent response as a result of H3.1. So in conclusion, we show that H3.1 inhibits Nef-mediated MHC downregulation in both primary CD4 positive cells and CD4 T cell lines. We also show that our transgenic mouse model is suitable to model an HIV/AIDS-like phenotype. We provide insight into how Nef inhibitors function as adjuvants by improving surface MHC1 expression, which is absolutely needed to combat Nef-mediated MHC class 1 downregulation that occurs during reservoir reactivation. And finally, Nef inhibitors are broadly applicable as adjuvants for DC-based vaccine or reactivation strategies discussed here, all the way up to vaccine strategies that consist of chimeric antigen receptor T cells, or CAR T cells. So with that, I would like to thank funding sources, lab members from University of Western Ontario, as well as collaborators and lab members from abroad. Thank you.